system of attraction. You've got to prove that. So here's some work in that direction. This is in Mathematica. I set up a simulation in Mathematica using nestlist, which does the recursive forward stepping that the Python also does. But there's a nicer thing you can do here. You can take a derivative of the value you're at with respect to the initial value, the value in your basin of attraction. And so if I can show that that derivative is 0, that means the end result is independent, and that would be, in my mind, a proof. Well, when I calculated it, dp is the amount by which p is changed when it's decremented by itself. So it's small, 0.01 is what I used. And q is like 3. So that's a small number taken away from 1. So I have a number between 0 and 1, and I raise it to a high power, 40 times 3, and that's going to be 0. Okay, that's still not a proof. But I claim that you will probably agree that the chain rule and an induction will be the proof.
Well, also, this is like just giving you an idea of the rate at which it approaches it.
And that too. And furthermore, why don't we ask Mathematica to solve the equation? Okay? That'll tell us everything. But here's the thing. I'm using n equals 2, so I'm doing octaves, just a frequency doubler. It says that p goes to 1/2 q, fine, times this thing, this horrible thing. I don't know what a product log is, but Mathematica knew how to integrate a rational function, vector field, and came up with this, and I said to myself, well, I don't like that. It better disappear as time goes on. So I evaluated it for a time out to 120 nanoseconds, whatever. And I came up with this number, which is very close to 0. That's not a proof, again, but I don't know how to prove things about product law. And I wouldn't want to do that these days. It would not be easy, because that's not just a product log, it's a product log of a very complicated function.
And P represents a...
That's the B, the timeout of B.
P is the timeout of P. B.
Yes.
Okay, got it. So that's actually a time then.
Yes.
Okay. So I'm trying to figure out what the number actually means. The number is like a...
This number?
Yeah.
Oh, well...
Is that would be...
That's
would that
the
be like
limit
a
of
unit
time
of
going
time?
to...
I'm sorry, oh, time going to...
Yes, that's it. In the limit of time
Yeah.
going to infinity, this term turns into 1.
Yeah.
This factor, I should say, turns into 1.
So I'm wondering if you integrated that with, like, let's say, I mean, we have absolute units for time. You have Planck time, right? There's the point of time where it's basically indistinguishable.
That's,
like
what I think this is telling us.
Okay. Or at least you can figure out how many steps it would take.
It would be nice to do that, yes.
Okay. Anyways, very interesting. Carry on.
Okay, so change gears, because there's still plenty of biology that I want to bring into the mix. This is a schematic of extreme simplicity of a cell, a biological cell. So it has an envelope, and there are 3 departments. The metabolic department has to do with nutrition and providing the energy for the cell to operate, to be alive. I'm not going to talk about that at all. It involves a lot of very small molecules. There's also the signaling part, which of course is extremely interesting because I am talking about dotted arrows. But I'm not going to talk much about that either, other than to say signaling has to do with binding of molecules external to the cell to macromolecules that are stuck through the membrane of the cell. And they know how to bind to some things and not others. And when they do bind, the interior part of the thing sticking through the wall of the cell wiggles some chemistry down inside the cell to make things happen. Most interestingly, from what I am aimed at, is something called the genetic regulatory network in the cell. Every cell has it. Every living cell has this very interesting interior mechanism, which is what the central dogma of biology is about. So I want to tell you about that a little. Oh, I told ChatGPT what I think it's about. I tried to summarize in one sentence the central dogma of biology, going from translation to, I should say, from transcription to translation. And ChatGPT, a machine, gave me approbation. And I love it when a machine tells me I'm okay. Now, you might think, as I do, that the stuff that mathematicians work on, such as spectral sequences and algebraic topology, or the really good stuff leading up to Blavatsky's unification axiom, that's hard for me, really hard. But, you know, take a look at a cell.
Well, and don't forget what Blavatsky himself said that.
Oh, did he?
Well, yes, he was interested in categorical biology also, and he.
Oh, I didn't know that.
Yes.
Good.
So then he did decide to go something simple like invent homotopy type theory instead.
Oh. Well, here's a cell in an extraordinarily simplified schematic form. And the main thing I'm interested in is called the ribosome. But before I get to that, I just want to mention Roger Penrose and a scientist named Stuart Hameroff in 1994 were giving talks at the first conference on the science of consciousness in Tucson, Arizona. And they were talking about microtubules, which are these things here, microtubules. And they said that's where consciousness is. That's where it is. And microtubules are actually just sort of like a subway system for moving molecules around inside the cell. I'm not going to talk about that. But ribosomes come up in the central dogma of biology big time. So that's what this chart is about. The central dogma says that the flow of information is unidirectional. It starts with DNA, which gets processed by something called RNA polymerase and a whole lot of other interesting things which change how RNA polymerase binds to the DNA to result in the production of an intermediate thing called messenger RNA, mRNA, which then finds itself out of the nucleus where all of this happened. This is called transcription. The mRNA finds itself out of the nucleus to a ribosome, which is this gadget here. I like to make an analogy. I don't know if you're familiar with a machine shop. Machine shop has tools like bandsaws, grinders, lathes, and milling machines. And I took a course in this in high school at Stuyvesant. Did you?
No, we have woodworking.
Yeah, woodworking.
Yeah.
Do they still have metal?
I doubt it. I don't.
Well, anyhow, in those days they had metal. And a milling machine is a fabulous tool. It's a big thing. And so is a ribosome. It's a big thing on a molecular scale. And you put in different cutters to make different shapes out of a block of metal. And you have cranks. Nowadays it's all automated. But anyhow, in my day, you did it by hand. You had to do it at the right speed. Went too fast, you would burn the metal. Went too slow, you wouldn't make any progress. So it was a skill. But anyhow, ribosome is a fantastic, complicated machine that takes the mRNA. Okay. It takes the mRNA and spits out a protein. That's the dotted bead at the bottom there. We are made out of proteins.
Yeah.
The whole thing is proteins. And the proteins are ultimately being produced as a result of the encoding in the DNA. Now, there are lots of ribosomes. When I showed you this picture of the cell, there were ribosomes, I don't mean that one. Okay, hold on. Yeah. These little black dots, these are all ribosomes. There's a lot of them, they're floating around. And they're all doing what I just said, in parallel. And there are a whole lot of them also attached to these green things. In this picture, this is a cross-section. And what that's really a cross-section of is the endoplasmic reticulum, which is a very interesting topological structure. As soon as I saw that, I said Morse theory. Because I'm thinking of a rising plane that's hitting the critical points here at the bottom and the inside. And this is just a, you know, as you can tell, it's just a cutaway view. The whole thing is like this. It's a round three-dimensional thing with multiple layers. And the endoplasmic reticulum has two surfaces. One surface, which is coated with ribosomes, is called the rough endoplasmic reticulum, and it has the smooth, no ribosomes. All of these ribosomes, both those floating around in the cytosol of the cell and those attached to the endoplasmic reticulum, are producing proteins. What happens to the proteins? Well, for one thing, they build more cells. But for another thing, they go back into the nucleus. So, let me just be clear about it. I had to check on this because I have not found any book that says what I'm about to say. There's a feedback loop between the DNA to the RNA, which goes out through nuclear pores to the ribosomes, which generate proteins, which go back into the nucleus through pores as well. It's bidirectional pores. Yeah.
I can't tell you a book, but I can tell you plenty of papers because it's exactly something I've been immersed in for a lot.
Can we talk?
Yeah, of course. Yeah, we'll have plenty of time afterwards.
Thank you. I look forward to that, for sure.
Venter and people like that, yeah. Speak by Venter and other people about exactly that.
That's very interesting. Thank you. So, in the cytosol, not in the cytosol, inside the nucleus, there are a bunch of small molecules called transcription factors. These are proteins that found their way back in to the inside the nucleus to either support or not support the binding of RNA polymerase to generate proteins according to the design built into the DNA. This is the DNA. And in front of each gene, there is a region in the DNA called the promoter region, to which various things can bind to produce a Boolean algebraic control of the production of proteins resulting from the mRNA. So I want to show you more about that. Here is the code. This is the genetic code. So
I just want to point out, just in terms of time, you're up to slide 50, and you're going to go all the way to 110?
Let's skip this. You know about the genetic code. Here's a ribosome, a picture of the ribosome. And here's paper has a finite state linear model of the genetic regulatory. Thank you. I knew this was going to happen. The Boolean is in the transcription factors over here. And in this model, what happens is the turning on and the turning off of two genes, these are the products of two genes, which have feedback to the Boolean inputs of themselves and to one another. In a very actual genetic regulatory networks have thousands of triangles and squares. Very complicated. But I wanted to make one with a pulse diagram, so there it is with two function rooms, a couple of states that are spinning, and here's my oscillograph. The red line is protein, and the black traces are the transcription factors that are turning on and off the production or not of a gene represented by P, which stands for protein. Here's a new topic. On the left, I have a pulse diagram. Here's the code for it. It's a frequency sweep. It's sweeping downward from a high frequency to a low frequency. And then it starts over again, because this module here is in a circle. When I was at the Rockefeller University, I was introduced to a very brilliant high school student, Matthew Elvey, and we had found in the magazine Science a biological model of the breathing of a snail, limnae. And so I had written a Java program to simulate pulse diagrams. So he translated that paper into pulse diagrams, and he gave a talk at Hunter High School. Okay, so we need a theory of pulses and a theory of pulse diagram, pulsations. So I needed to take advantage of something that James Allen came up with, was an analysis of the possible relationships between pulses as intervals. So he's not talking about pulses. He's just talking about the intervals of the pulses. But he's not talking about pulsations in the sense of a whole series of them, nor is he talking about parallel series of them. So here's some pictures of parallel series of pulses. Here's a first order theory that's in the literature about those things. I can try to, this is a sketch, but my main point is this, is number 7. This says that each state has a set of pulses associated with it, such as like the generator A, or any other possibly controllable or retriggerable state. So, in addition to that, I threw in a monoid of time duration, a commutative monoid of time duration, so that every interval has a duration. Mind you, I don't mean necessarily a numerical real number, and that's a distinction I want to make. I call that the standard interpretation. If the duration of a pulse or an interval is in the real numbers, then of course you can take the, now you need more than a monoid, you need the group, because you want to do subtraction of the endpoints to get the duration. And this is where I now revert to a previous remark. I'm trying to think of mental time, phenomenologically, as being a bunch of pulse diagrams that are built into your neurons. Just sitting there. If I talk to you about baseball, for example, that doesn't do anything unless you're familiar with baseball. If you are familiar with baseball, I would ask you, I'm doing a thought experiment, imagine a fastball pitch. Did you do it? Now, 90 feet from the pitcher's mound to the home plate at 90 miles an hour, the fastball pitch is about a little over half a second, I think. But when you imagine it, it was over a lot faster than that. So there are 3 times involved in that thought experiment. There's the mental model of baseball pitching, there's the imaginative simulation of it, and there's the actual pitch. And so I just want to remind you, I said this earlier down there. Okay? Going back to Lotka-Volterra, I can do this in 10 minutes. Is that okay?
Yeah, sure.
Because I got to get to con extensions.
You got to get to Rosen. Rosen. The life.
Robert Rosen.
Okay, now I'm not going to talk about Robert Rosen's book. I read that book and I had a lot of problems with it.
Well, there are papers of his where he was dealing with delays in his MR business. That
Yeah,
part, that part is
Let's focus.
Yeah.
Let's think. Then we could talk about it forever.
Okay, so Ernst Mayr, one of the grandfathers of the modern dogma, pointed out that the Lotka-Volterra story is historically important from ecology. Now I'm going to Michael Barr, because Lotka and Volterra came up with those equations independently of each other a couple of decades apart, for different reasons. One was Lotka was working on biology, but Volterra was working on fish populations during the First World War. And they both came up with the same set of equations. Well, here's a little story, side story for you, mentioning Michael Barr. Eilenberg was attending a talk by MacLean in which MacLean was showing a calculation, and Eilenberg recognized it. That was the beginning of category theory. That was when they started working together on homology and cohomology of groups. Same equation in two different areas of research. Same thing. I find that to be really stimulating. Okay, so the Lotka-Volterra equations produce two pulses, the rabbit population and the fox population. So you got these two populations. Of course, you want to see that as a phase diagram so that you can see the basin of attraction and the cycles of attraction. So, there's that. And Brendan Fong and David Myers and David Spivak at MIT, currently top-notch applied category theorists at MIT, have a paper on behavioral mereology, which is parts and wholes, and they use, basically their work is based on Lauvier's categorical logic, as I will show very briefly. But the point is one of their running examples in that paper is also about foxes and rabbits. Now, there's a category theory paper about foxes and rabbits, and I'm not going to open it up, but they show a general form of the Lotka-Volterra, a very general form, and they have a Lotka-Volterra functor. So, you know, it has reached category theory. It's a beautiful paper, by the way. But here's the pulse diagram for Lotka-Volterra equations. Got a couple of three function rooms. Got a dependent variable, which is the product of the population of foxes and the population of rabbits, and this thing models. So here's a case where continuous differential equations are becoming a discrete model, as opposed to what I mentioned earlier, where when I saw Brian Scrumpquist's discrete model of a frequency doubler, I wrote a differential equation. It's in the opposite direction. Now we get to stochastic chemistry, so I'm going to really go very fast. But this paper in 1977 by Dan Gillespie had over 12,000 citations cumulative until recently. What he did was he invented an algorithm for simulating chemical reactions that is based on a stochastic approach rather than the law of mass action, which was standard until him. So I took his mathematics and rewrote it in modern notation with stochastic processes. But his main contribution was the invention of propensity for a reaction to occur. Propensity is a number, A mu x0, which depends on the concentration of the chemicals in the vat or the cell. And that number is a probability per unit time. He invented that. And So what this equation says is that, by assumption, this is a postulate, the probability of reaction mu occurring by time, in the time delta t, given the concentration x arrow, is a mu times delta t. Which, by the way, is just sort of a variable Poisson process. Because in a Poisson process, you got lambda delta t, where lambda is the parameter of the Poisson process. So I wrote that up, and here's the theorem, which says that the probability of the next mu reaction after a waiting time, t, within the next tiny amount of time, delta t, is given by the product of a continuous probability distribution and a discrete probability distribution. Again, this theme in mathematics of the discrete and the continuous is always with us. So I proved that theorem with this calculation, and then, there it is, and then I took his flow chart, that's from the 1977 paper, and made a pulse diagram out of it, which has this module here. That's what does the time, the waiting until the next reaction. And there's a theorem to prove that, which is in a book that I wrote. So I don't have time to go over that. But this thing here has the stochastic.
What's the name of the book?
Microlects of mental models.
Okay. Microlects of mental models. Okay.
I'm not going to talk about that. I wish I could.
Okay, now it's on the internet, so
It's on
people can
Amazon.
look it up.
It's on Amazon.
Oh yeah, I know it's on Amazon. I'm saying this will be on the internet, so
Yeah.
You watch
rest
on
of the world
video.
will know about it.
And by the way, a PDF of that can also be purchased on our website, but never mind. This part here is the interesting part, because that's where which reaction occurs is chosen by taking the propensity of each of the possible mu reactions and dividing by the sum of all the propensities, so you get probabilities, and that's the probability arrows that I want to use. And then nu here is the chemical reaction. Nu is what you subtract from the reactants and what you add to the products in any normal chemical reaction formula. Okay, can I do this?
Yeah, go.
'Cause
Anybody who wants could leave, whatever. But at some point, the camera's gonna die. But
Oh, well,
his
his camera won't.
Okay.
Anyhow, I really want to show you, I'm changing gears now, because I'm going to change to the finale, which is about cellular automata. Because cellular automata correspond to left Kohn extensions. And that's a theorem in a beautiful piece of work, which I want to tell you about.
Definitely want to hear it.
And then connect it almost to pulse diagrams, and that's the talk. All right, so I got to show you what a cellular automata looks like.
Go for it.
So I'm going to just turn one on here. I want larger than life. Larger than life. Bugs. Simple rules can create complicated phenomena. That's Stephen Wolfram's mantra for life and the universe. He wants to find one equation for the entire universe. You've heard of that.
Yeah, I
He
read
wrote a
his
book called A New Kind of Science. It's based on
Yeah,
this.
great book.
You like it?
Excellent.
I
Yeah,
tore it apart.
well, I didn't read the whole thing, but lots of pictures in it. But it was
Lots of pictures.
It was
So-
phenomenal work.
So I just want to observe that the homology of this little creature changes over and over again. Okay, so there's that, but now I want to show you a different one, just to speed things up a little bit. I want to talk about one-dimensional ones, 1D binary. And I made this one for this talk. There's only one active cell initially. But when you run a certain rule,
Mhm.
you get that pattern.
Yeah.
And I want to tell you more about that. Please.